In grade 8 science, you learned that cells divide to produce new cells, and meiosis is one of the processes producing genetic variations in Mendelian patterns of inheritance. Today, in grade 9 science, you will focus on describing the location of genes in chromosomes and explain the different patterns of non-Mendelian inheritance. So without further ado, let's get started. Before we proceed, let us start with unlocking of difficulties. These are the terms that you will encounter all throughout the lesson, so let us define them and be familiar with this. First term. It is a branch of biology concerned with the study of genes, genetic variation, and heredity in organisms. This is genetics. That's right. The next term, a process by which genetic information is passed on from parents to child, that is inheritance. Any difference between cells, individual organisms, or groups of organisms of any species, that is variation. Next, a unit of heredity, a section of DNA that codes for a specific trait, that is our gene. Next, I know that you are all familiar with genotype and phenotype. Now let us differentiate them. First up, genotype. It is the genetic makeup of organism. It is the combination of two alleles. While the phenotype is the physical appearance or the physical feature or the trait of an organism. Let's have an example of genotype and phenotype. For a clocked flower, so I have here two flowers as you can see. What is the genotype of the first flower? The genotype is FR, the combination of two alleles. How about the other one? That is FW. That is the genotype, the genetic makeup of organisms. While the phenotype of the first one is red because it is color red, it is its appearance, and the other one is white, the physical appearance or the feature or the trait of the organism. That is the difference between the genotype and the phenotype. Is that clear, class? Now let us proceed. Let us define homozygous. It means having two identical alleles, and heterozygous means having two different alleles. Next up, I know that you already know how to use Punnett square. Punnett square is a graphical representation of the possible genotypes of an offspring arising from a particular cross or breeding event. Reginald Punnett is the proponent of Punnett square. Next, do you know Gregor Mendel? Gregor Mendel began a decade-long research project to investigate patterns of inheritance. He studied the inheritance of 7 different features in peas, including the height, flower color, seed color, and seed shape. Gregor Mendel's principles form the base for the understanding of heredity and variation. That is why he is considered the father of genetics. Next, non-Mendelian inheritance. This is a type of inheritance wherein the patterns of phenotypes does not coincide with those that was presented in the Mendelian law of inheritance. And this is our lesson for today. The 3 types of non-Mendelian inheritance. Let's have the first type. That is incomplete dominance. It is a form of intermediate inheritance in which one allele does not completely dominate another allele, resulting in a new or a third phenotype. Let's have an example of incomplete dominance. We have here a red snapdragon flower crossed to a white snapdragon flower, and the result is a pink snapdragon flower. One allele does not completely dominate another allele, resulting in a third or new phenotype, and that is the pink snapdragon flower. Now, let me show you how it happened using the Punnett square. As you can see, our genotype for red snapdragon flower is FR-FR. Our genotype for white snapdragon flower is FW-FW. Let us cross using Punnett square. So this is our genotype. Let us cross it downward and then sideward. So these are the results. Now let us identify the genotypes. Always remember each box is equal to 25%. So as you can see, they are all the same. All the genotypes are the same, FR, FW. Therefore, 100% the genotypes are FR, FW. The phenotype is the physical appearance. Now, the result when we cross red snapdragon flower and white snapdragon flower is 100% pink snapdragon flower. That is how we use Punnett square. Let's have another example. Show the possible outcome of the cross between white flower and pink flower. So what are our genotypes? For white flower, our genotypes are FW and FW. For pink flower, our genotypes are FR and FW, not FP and FP. That is wrong because pink flower originated from the red and white flower. That is why the genotypes are FR and FW. Always remember that. Now let us cross FW or the white flower and the pink flower using Punnett square. Downward and then sideward. So here it is. As you can see, we have here 2 FR-FW and 2 FW-FW. Now let us determine the genotypes and the phenotypes. The genotypes are 50% FR-FW, 50% FW-FW. How about the phenotypes? What is the meaning of FR-FW? That is pink flower. Therefore, we have 50% pink flower. How about FW-FW? It means white flower. That is why our phenotypes are 50% white flower and 50% pink flower. Understood, class? Now let us proceed to the second type of non-Mendelian inheritance, and that is what we called codominance. Codominance means both alleles are expressed equally in the phenotype of the heterozygote. For example, red flower plus white flower, the result is red and white flower. As you can see, in codominance, both alleles, both appearance, or both traits are expressed equally, red and white. That is codominance. Let us have an example. Let us cross red and white flower. So here it is, as you can see in using Punnett square. 100% red and white flower. Now let us have an activity. I will be showing pictures and you are going to distinguish if it is incomplete dominance or codominance. Let's get started. First one. Standard poodle plus Labrador equals Labradoodle. What is this? Is this incomplete dominance or codominance? This is incomplete dominance because one allele does not completely dominate another allele, resulting to a new phenotype. That's right. Next, we have here white chicken mated to black chicken. The result is checkered chicken. As you can see, what is this type of inheritance? This is what we call, that's right, codominance. Because both alleles are expressed equally. As you can see in the chicken, it has white and black. Next, orange fur cat and black fur cat. What do you think is the result? Orange and black fur cat. And this is an example of codominance. That is right, because both alleles are expressed equally. Okay, next one. We have here straight hair and a curly hair. What do you think will be the result? We have wavy hair, and this is an example of incomplete dominance because we have a third phenotype or a new phenotype, which is wavy hair. Okay, good job. I know that you already know the difference between incomplete dominance and codominance. Now let us proceed to the third type of non-Mendelian pattern of inheritance, and that is what we call multiple alleles. From the word itself, multiple, it is a gene that is controlled by more than two alleles. And one good example of multiple alleles is our blood type. Do you know your blood type, class? It is important that we know our blood type. Okay, so we have 4 blood types. We have blood type A, blood type B, blood type AB, and blood type O. So which blood type do you have? These are the 4 blood types. Blood type A has antigen A, blood type B has antigen B, blood type AB has antigen A and B, and blood type O has no antigen or neither antigen A nor antigen B. Next, so these are the genotype of blood types. For blood type A, as you can see, we have 2 genotypes. We have the homozygous IA and the heterozygous IA small I. Blood type A can receive blood from itself, blood type A, and blood type O. Blood type B, we also have 2 genotypes, homozygous and heterozygous. Blood type B can receive blood from blood type B and blood type O. For blood type AB, it is an example of both codominance and a trait with multiple alleles. It has a genotype of IA and IB. It can receive blood from blood type A, blood type B, blood type AB, and blood type O. That is why it is called the universal blood receiver. Blood type O has a genotype of small I, because it has no antigen. It can receive blood from blood type O only. And as you can see, blood type O can donate blood to any type of blood type because it is considered the universal donor. Now let us have a problem. A couple gives birth to a baby boy. Both parents have blood type A. However, there is a confusion in the hospital. There are 2 babies which have unknown parents. The first baby has blood type B, while the second baby has blood type O. Help the couple find their son. So for this problem, you are going to answer this on your paper. You will construct a Punnett square to solve this problem. For blood type A, we have 2 kinds of genotype, the homozygous and the heterozygous. According to the problem, there is no specific detail that the parents are homozygous or heterozygous. That is why we are going to do a trial and error. Trial number 1, both parents have homozygous blood type A. You will cross this using Punnett square. For trial 2, one parent is homozygous and the other one is heterozygous. For trial 3, both of them are heterozygous. Let us see which one is their real baby. These are the guide questions. List down the genotypes and the phenotypes for each trial. And for question number 2, who is the real son of the couple?